in this session, we will be talking about the reproductive biology of angiosperms. Angiosperms have a characteristic life cycle in which there is an alternation of generation between a diploid sporophyte and a gametophyte and a haploid gametophyte. The gametophyte is totally dependent on the sporophyte for its growth and development. Male gametophyte is a pollen grain, and today we will be talking about the anther. Now, a typical flower consists of 4 whorls of leaf-like structures. Basically, flower is a structure which is involved in the reproduction of angiosperms. It is a determinate shoot and it comprises of 4 whorls of leaves. Starting from the outside, these whorls are the sepals, the petals, the androecium, and the gynoecium, or we say the outermost whorl is a calyx which comprises of sepals. Inner to calyx is the petal, and petal it comprises of the calyx, and then there are 2 reproductive whorls, that is the androecium, the male reproductive structure, and the gynoecium, which is the female reproductive structure. Now, in this particular slide, you can see that the outermost whorl of leaves on the receptacle is the calyx. Inner to calyx is present the corolla, and then inner to corolla, the 3rd whorl is the androecium, which comprises of the stamens. These are the male reproductive structures. The innermost or the 4th whorl is gynoecium, also called as the pistil, which is the female reproductive structure. Now, most flowers have all the 4 leaf whorls present in them, and we call them as complete flowers. Those flowers in which any of the whorls is missing, those flowers are referred to as incomplete flowers. Some flowers may have just the male reproductive structure, and these flowers are called as the staminate flowers, whereas those flowers where there is only the female reproductive structure, those flowers are called as the pistillate flowers. So, we can have 3 types of flowers, that is flowers in which both the sex organs are present. Such flowers are called as the bisexual flowers. And then there are unisexual flowers, which could be either staminate flowers or these can be pistillate flowers. There is a huge diversity of flowers which exists in angiosperms. As you would have learnt in your earlier lessons, flowers can be present singly on a shoot, or they can be present in an inflorescence. The largest inflorescence in angiosperms is of Amorphophallus, which is about 3 meters in length. The smallest flower is of Wolffia, which is very, very minute, and it is microscopic because it is about 0.1 millimeter in diameter. In contrast, the largest flower is of Rafflesia, whose diameter is about 1 meter. In this particular slide, you can see a whole range of diversity of flowers. They are of different colors, they are of different sizes, and of different shapes. Now, the color and the shape of the flower is very instrumental in the reproduction of any particular species, because the pollinator is also decided on the basis of the color, size, and shape of the flower. Besides this, there is also diversity in terms of nectar and odor, which is emanated by the flowers. Now, like I told you that in this particular class, we will be talking about the reproductive biology. So, we start with the male reproductive structure, which is a stamen. Now, stamen consists of 2 parts, that is an anther and a filament. Anther is a lobed structure, and this lobed structure is borne by an elongated filament. Filament is attached to the receptacle, and it is by means of this filament that there is conduction of water and nutrients to the anther. Anther contains the pollen grains, and of course at maturity the anther dehisces, which leads to the dispersal of pollen. So anther is basically the site of pollen development and dispersal, and pollen grains are the carriers of male gametes, or we also say that pollen grain is the male gametophyte which carries the two male gametes. We also refer to these male gametes as the sperm cells. The filament of the anther is also instrumental in positioning of the anther. If the filament is small, then the anther is very much close to the receptacle, but if the filament is elongated and if by chance it is facing towards the outside, then this is important in the dispersal of the pollen grains. Talking about the anther, it is a bilobed structure and it is a ditheca structure. Theca is basically, theca we refer to as the cavity of the anther lobe. So each anther lobe has one cavity, and each cavity has two microsporangia, making an anther a tetrasporangiate structure. So if there are two lobes and in each lobe there are two microsporangia, in all there are four microsporangia. So anther becomes a tetrasporangiate structure. Now the two lobes of the anther, they are joined by a connective, and this connective is also attached to the filament. In each lobe there are two microsporangia which are present, and these microsporangia are separated by a strip of sterile tissue which basically arises from the epidermis, and this sterile tissue of the epidermis is later on instrumental in the dehiscence of anther, and this sterile tissue is called as the septum. Now, I just mentioned that an anther is a bilobed and a ditheca structure, but there are certain anthers in which the two microsporangia in a theca, they get fused to form a single theca. In those cases, the anthers are no longer bilobed, they are monothecous anthers, and these are commonly seen in Moringa and in Wolffia. This anther basically comprises of both types of tissues, that is sporophytic as well as the gametophytic tissue. Now we will talk about the development of anther. Basically, anther develops from a homogeneous mass of tissue which is an undifferentiated anther, and this homogeneous mass of tissue is present within the epidermis. Epidermis is the outermost layer, it is protective in function, and there are some groups of cells which are present inside the epidermis, that is, they are hypodermal in origin, which form the archisporium. Now, archisporium can be a single cell, or archisporium can be a multi-layered structure of cells which are vertical in position, and this archisporium basically is distinct by elongated cells which have a dense cytoplasm and they have a prominent nucleus present in them. The archisporial cells, they divide transversely, forming a parietal cell on the outside and a sporogenous cell towards the inside. Now all the wall layers of the anther, they arise from the primary parietal cells. Primary parietal cells can undergo mitotic divisions and give rise to secondary parietal cells, which will eventually form the other wall layers apart from the epidermis. Epidermis is the outermost wall layer of the anther. So there are 3 more wall layers, that is the endothecium, the middle layers and the tapetum. So they all arise from the primary parietal cells. On the other hand, the archisporium has also given rise to primary sporogenous cell, which is present towards the locule of the anther. These are the primary sporogenous cells. So primary sporogenous cells can directly function as the microspore mother cell, or they can undergo a few more mitotic divisions, increasing the number of sporogenous cells, and each of these cells will now function as the microspore mother cell. So eventually meiosis will take place in the microspore mother cell, and these microspore mother cells, after undergoing meiosis, will form the microspores, which will subsequently transform into the pollen grains. So I would like to repeat here that in an undifferentiated anther, the outermost layer is the epidermis, and then inner to epidermis, the hypodermal layer is of cells which comprise the archisporium. Archisporium cells divide transversely, forming parietal cell on the outside and sporogenous cell on the inside. Sporogenous cells give rise to the microspore mother cells. The primary parietal cells, they can divide and form secondary parietal cells, and they give rise to the rest of the wall layers of the anther. Now here in this picture, you can see the transverse section of an anther. Now the A part of this diagram shows one particular stamen with the anther and a filament, and you can see that it has been cut in a transverse manner. So when you cut a transverse section of an anther, so you see what you see in B portion of this picture. There are 4 round structure which you see here. These are the 4 microsporangia which harbor the spore mother cells, which will divide meiotically and give rise to the microspores and the pollen grains. Right? And in the center, the darkly staining structure which you see is the connective, that is the point of attachment of the filament to the anther. Okay, so this is basically, this is present between the 2 anther lobes, as you can see on the left hand side in figure A, and each of these 4 microsporangia, they are enclosed by 4 different wall layers. Now, you can also see that there is a very prominent stomium present between 2 microsporangia on each side of the anther. This is the region where the cells will dehydrate and eventually the dehiscence of anther will take place. Dehiscence of anther is important because in a mature anther, these microspores are converted into pollen grains, and pollen grains or the male gametophytes are the carriers of the male gametes, which eventually are important in fertilization. They carry out the double fertilization. There are 2 male gametes which are present. Now, in the C part of this figure, you can see that there is epidermis towards the outside, that is the outermost wall layer is epidermis. Inner to epidermis is a single row of cells which are elongated. It is the endothecium. It is followed by 2 to 3 middle layers, and the innermost wall layer is tapetum. Now, all these are the wall layers of the anther. Inner to tapetum, you see that there is sporogenous cavity which harbors the spore mother cells, which eventually undergo meiotic divisions and they form the microspores. Spore mother cells are also called as the microspore mother cells, and they are also referred to as the pollen mother cells. Now, this picture is of a mature anther. You see that as the anther grows, with the growth and development, the volume of the anther increases and the epidermis has to increase accordingly because it has to cope up with the enlarging mass and volume of the anther. The outermost layer, which is a little stretched, is the epidermis. Then you see the endothecium. And then in this particular one, the 2 to 3 middle layers are not very pronounced because middle layers are ephemeral, as I will be talking to you very shortly about them. And in the microsporangia, you see that there are mature pollen grains which are present. Microspore has one nucleus, and when this microspore undergoes a mitotic division, there are 2 nuclei present, and here in this slide you can see bicelled pollen which are present. And towards the right corner, you can see that there is a pronounced stomium of elongated and dehydrated cells which play a role in the dehiscence of the anther. It is very important for an anther to dehisce at the right time, because in case the dehiscence is either delayed or it takes place before the maturity of the anther, then it leads to the sterility of the species, because the pollen grains of the right maturity are not delivered. Now, this is generally a summary of all the cell types which we come across in an anther. Like I just told you, connective is primarily, it is joining the anther theca together, and it also is supporting, it is giving a support to the anther. Epidermis is like our skin, which is the outermost layer. It prevents the loss of water, it facilitates the exchange of gases, and to some extent is also involved in the dehiscence of the anther. Then there is stomium, like I just told you a few minutes back. This stomium has a role in the dehiscence of anther. Then endothecium is a layer which is primarily responsible for dehiscence of anther, though it does perform a little bit of function of the support. There are 2 to 4 middle layers which are present, which primarily are involved in storage and are also involved in dehiscence of anther. The innermost wall layer is tapetum. Tapetum is the most important wall layer which is involved in the nutrition of the pollen, the growth, development of the pollen, and also involved in the wall formation of the pollen, or we can say that it is involved in the pollen wall synthesis. Now, inner to the wall layers, there are sporogenous cells, and these are instrumental in the formation of male gametophyte or the pollen grain. And of course, there is vascular bundle, which is important for connection and nutrient supply from the mother plant. Though tapetum, I told you, is a layer which is involved in the nutrition of the developing microspores, but this nutrition is coming from the mother plant, and it is the vascular bundle which is a connection which is supplying the nutrition from the mother plant to the sporogenous cavity through the tapetum. Now, we will take all the layers of the anther wall one by one. The outermost or the first layer of the anther wall is the epidermis. This epidermis comprises of columnar cells. It encloses the archisporial cell, like I just told you in the last 2 slides, and it is protective in function. Epidermis also provides structural support to the anther, and it plays a role in anther dehiscence. But the primary role of this epidermis is protection. Apart from that, it also prevents loss of water from the anther. It helps in the gaseous diffusion, and as the anther matures and enlarges, this layer becomes stretched and flattened in a mature anther. Inner to the epidermis, or The second wall layer of the anther is again a single layered structure which is hypodermal in origin, and it is called as the endothecium. The cells of the endothecium are generally uninucleate and they are highly vacuolated. They are somewhat elongated. They are hygroscopic in nature, and the characteristic feature of endothecium is that there are present some thickenings of alpha cellulose which are present on the inner tangential walls of the endothelial cells. These thickenings, they run outwards and upwards from the inner tangential walls. The fibrous bands here, along with the stomium, they provide a mechanical force which is required for the dehiscence of anther. In case there is a disruption in the thickenings, this leads to the dehiscence of anther being arrested, and eventually it causes the sterility in the male which is present. Now, in this particular slide, you see that there is an arrowhead which points towards these cells. You can see pronounced endothelial wall thickenings. You can see that it is a single layer of cells, and these are the thickenings, and these are very important for the dehiscence of the anther. So, the outermost layer is the epidermis, followed by epidermis is the endothecium, and then inner to the endothecium we have middle layers which are usually ephemeral. By ephemeral I mean that they are short-lived. So, there can be 1 to 3 middle layers present in different species of angiosperms. So, but there are certain species where there are no middle layers present. Polypleurum, vallisneria, wolffia, there are some of the species where there are no middle layers present. Now, the cells of the middle layers are very much flattened, they are thin walled, they are uninucleate, and they are vacuolated. They are not really pronounced. Sometimes the cells can develop fibrous thickenings. They can store reserve food material like starch, which can be later utilized during the growth and development of the pollen grains. And these middle layers, they play a role in the maturation of pollen, anther dehiscence, and to some extent also in the elongation of the filament. But when we look at the mature anther, like I had showed you earlier, we can go back and we can see that these layers here, you do not see any middle layers in this mature anther, because they get crushed. They get crushed in mature anthers. Now, ranunculus is one species where these middle layers are persistent. The innermost wall layer of the anther is the tapetum, and this is the most important wall layer of the anther. Tapetum is usually single layered, and it too, like all other wall layers, is derived from the parietal cells. Now, tapetum is the innermost layer of the anther wall, and it completely surrounds the sporogenous tissue. This is high in metabolic activity. Rarely it arises from the connective. Like I said, that it arises from the parietal cells, but sometimes it can arise from connective, and sometimes it can even originate from the sporogenous cells. In Antirrhinum and impatiens, it originates from the sporogenous cells. Okay. Now, Electra thomsoni is an example where this tapetum is dimorphic. That is, tapetum is comprising of cells of two different morphology. Those cells which arise from the connective, that is inner towards the anther locule, they arise from the connective. These are large, these are elongated, and those cells which basically they arise from the parietal cells, they are small and isodiametric, giving a dimorphic appearance to the tapetum in Electra thomsoni. We will finish here and start again in the next lecture. Thank you.